Assalamu alaikum class. This is in this lecture we will learn about the topical MCQs, and we will cover the chapter number 1, which is the self studies, and here we will learn about the MCQs which are discussed, which were given in the past papers, right? So here is MCQ number 1. The diagram shows an eyepiece reticule and cell viewed through a microscope. When the eyepiece reticule was calibrated, and they covered the 12 divisions of the micrometer scale. There were 100 divisions in 10 nanometer of the scale stage micrometer, right? So here we have to find out the size of that particular cell which has been viewed through the micrometer, right? So what is the actual length of the cell? Whenever we find out that, so if we have to find out what that particular formula, calibration factor is equals to number of stage divisions. How many stage divisions we have in this 12 stage divisions, and how many conchular divisions means the division of the eyepiece, 32 divisions. As we can read here, from here the 10, 20, 30 and the 2 more 32 divisions. This will give us the calculation in millimeter, and we have to find out in micrometer. So we will divide it with 1000, and here comes out to be approximately 375 micrometer, right? So the most nearest answer here is the C. So it is the actual length of the cell will be 360 micrometer. So come towards the next MCQ. The photomicrograph shows the ultrastructure of a part of a cell. As we can see in the picture, we can see here the chloroplast. These are the chloroplast cells which are present in the plant cells. So what statement is correct about the type of the cell shows in the photomicrograph is correct. It is a plant cell because it has both chloroplast and a nucleus, but we can't see any nucleus here. Okay, so it goes wrong. It is a plant cell because it has chloroplast. Yes, it can be. It is an animal cell, so we will correct it from here. Okay, it is not animal cell, it is just a plant cell. So the option number B is correct here. MCQ number 3. A student examined a slide of the human blood with a light microscope and made a careful drawing of the different cell types. The student used an eyepiece reticule so that the relative sizes of the different cell types were drawn accurately. So in the drawing, red blood cells were 7 mm in diameter, lymphocytes were 6 mm, neutrophils were 14 mm in diameter. What is the linear magnification of that drawing? Means how much magnify the what was the magnification power of that particular microscope through which we have determined these results. So when we see, it can be about 1000 times magnification which give these results, because 100 times, 40 times, 10 times magnifications will not give these results. They are lower for that particular measurements. Now next MCQ is the diagram here shows a graduated slide, right, with a divisions of 0.1 millimeter viewed using an eyepiece reticule. This particular smaller division is 0.1 millimeter. 0.1 millimeter. Now pollen grains were grown in a sugar solution and viewed under the eyepiece reticule. Diagram 1 shows that the pollen grains at the start. Diagram 2 shows the pollen grains after 4 hours. So what is the growth rate of the pollen tubes? Pollen tubes growth rate is very low because it takes time, and we always measure the we always make the measurements in the micrometer, so it can be 5 micrometer per hour. What is the definition of the resolution of a light microscope? The degree of the sharpness produced by the microscope? No. Resolution means how much it can magnify a particular distance between the two subjects. The greatest distance between the two objects visible in the same field of the view? Yes, it can be. So the option number B is correct here. The electron microscope shows a type of the virus at a magnification of 30,000. What is the length of the virus? So the length of the virus comes out to be 2.2 and 10 is about 3 nanometer. Whenever we have to use the formula here, magnification is equals to image size by actual size. So we can use a calculation here, and we find out the 2.2 into 10 is about 3 nanometer size of the virus. A student was given a photomicrograph of the cell and told the magnification of the image. The student was asked to calculate the actual size of the cell. Which row in the table explains how to do this? So there are the 3 columns measuring the image. In which particular unit we will measure the image? Either is a centimeter or millimeter, or convert to micron meter by multiply with. If it is a millimeter, then how we will convert it into the micron meter, and if it is a centimeter, then how will we do it? So we have to find out all the things, and we have to relate that particular option which is the correct in all the cases. So, and I have read all the options here, and I found out the option number C is correct, because we always measure the image in millimeter and then convert it into the micrometer by simply multiplying it with 10 raised to the power 3. Rearrange the formula. The magnification formula is always image by. When we have to find out the actual size, then we have to magnification is equals to image by actual size. So whenever we have to find out the actual size, we do this. So number C is correct here. So now move towards the next MCQ. Which row shows the correct order of the size of these cell structures? Mitochondria, ribosome, cell surface membrane, and the chloroplast. Whenever we view all these, then we come out to be that the option number C is correct here, because the larger size is that of the chloroplast, then that of the mitochondria, and then ribosome, and then cell surface membrane. Question number 9. The diagram shows a stage micrometer scale viewed through an eyepiece containing the graticule. The small division of the stage micrometer scale is 0.1 millimeter. Stage micrometer scale have 0.1 millimeter as the smallest division. Means this division is 0.1 millimeter. Right. The stage micrometer scale is replaced by the slide of a plant cell. So what is the actual length of the nucleus in the plant cell? So if we will find out, then we come here that the actual length of that particular plant cell is 25 micrometer. The size of a measles virus, Morbilli virus is approximately 150 nanometer, and the Mimi virus is approximately 4.5 times larger than that of the Morbilli virus, whilst the Pandovirus is larger than that Mimi virus as well. So which virus can be seen using a light microscope with a maximum resolution of 0.25 micrometer and using an electron microscope? So I have read all the options and find out that the option number B is correct here, because the Morbilli virus is the smallest, so it cannot be seen with the light microscope, and we have to use the electron microscope. The Mimi virus can be seen and Pandovirus can be seen, so the option number B is correct here. The electron micrograph shows the type of the virus at a magnification of 60,000. So what is the actual length of the virus? So we have to find out that particular magnification is equals to image size by actual size. So we will use that particular formula, and then we can find out that the actual size of the typical virus comes out to be 1100 nanometer. What are the appropriate units for measuring the diameters of the alveoli, diameters of the white blood cells, and the diameters of the cell walls? Alveoli are too small, right? And the white blood cells are comparatively, they are also very small, and the width of the cell wall is also very small. So we have to find out which particular diameter which particular unit we will use to measure that diameters. So I have read all the options and I find out that the option number D is correct here. Whenever we have to measure the alveoli, we use micron meter. For white blood cells, we use micron meter because they are too much small. And for the cell walls, they are smaller than that, and we have to use the more smaller unit, that is the nanometer, right? The actual diameter of the prokaryotic cell is 0.5 micron meter. An electron micrograph of the cell has a magnification of 50,000. So what is the diameter of the cell in the image? So we will use that particular formula. Magnification is equals to image by actual size. Magnification here is 50,000 multiplied with 0.5, and image size we have to find out. The whenever we will calculate, it comes out to be, see, it comes out to be 2.5 into 10 raised to the power 1 millimeter. So option number C is correct here. The diagram below was drawn from an electron micrograph of an animal cell, right? Our diagram would represent the same cell under the same light microscope, using simple light microscope, using the daylight as the only source. As the daylight has a very low amount of the wavelength, so it has very low, it can it has very low resolution power, right? So we have to, so it cannot show the structures, more structures of the cell, for example endoplasmic reticulum, ribosomes, we can see in the other pictures. So option number A is correct here. It can show only a few things about, because it can show only a few things about that particular one. So option number A is correct here. The diameter of a red blood cell in a diagram was measured to be 2.5 centimeter. The actual diameter was 7 micron meter. So which calculation would give the correct magnification of the red blood cells in the diagram? So we have to find out magnification is equals to image size by actual size. So image size here is 2.5 centimeter by 7 micron meter. Whenever we will calculate, we just make the two sizes in the same unit, that is a micron meter. So it comes out to be 25,000 by 7, 25,000 by 7. So for the MCQ number 15, the option number C is correct. The diagram shows a fin wheel drawn to scale, which is a 3 meter. A student made 3 statements about the diagram. The magnification is 0.006 times. The ratio of the actual size to diagram size is 6166 and whatever. So we have to consider all the 3 points and find out which one is the correct. So option number C is correct here, because we find out that magnification is 0.006 and the fin wheel has an actual length of 24 meter, then that particular statements will be correct. So option number C is correct here. A student was told that the actual length of a cell structure is 5 micron meter. The student was asked to state an equation that can be used to calculate the magnification of an electron micrograph of the cell structure. The student used some of the letters Q to U in the equation. So Q states the length of the cell structure image on the micrograph in the centimeters, R in length of the image on the micrograph in millimeters, S is the 1000, T is 1 by 5, and U is, so when we used to calculate that particular magnification, we find out that it comes out to be R into S into T. So option number D is correct here. The actual length of a cell structure is 8 micron meter. Which steps are used to calculate the magnification of electron micrograph of the cell structure? Measure the length of the cell structure image on the micrograph in the centimeters. No, we have to measure it in the millimeters. So option number 2 is correct. And divide the image by the length by the 1000. We have to convert it into the micrometer. So we will not divide. We will multiply it with the 1000. So it is correct. Step divide by 8 because it is the 8 micrometer. So we will divide it by 8. If we are dividing, then we can multiply it. So option number D is correct here because it shows that steps 2, 4 and 5 are correct. What is the typical resolution of a microscope using the daylight as a light source of a 10x eyepiece? It comes out to be 200 nanometer. We have to remind it simply. 200 nanometer, right? Which combination of the lenses of a light microscope will give the greatest magnification? I have read all the combinations and I found out that this particular combination is the correct one. 15x eyepiece lens and 100x objective lens, then will be the greatest resolution. The photomicrograph shows the some mesophil tissues from the dicotyledonous leaf. These are the mesophil tissues which we can see there. So they comes out to be 5 micrometer in the diameter. What is the magnification of the photomicrograph? It comes out to be 2800 times. When we magnify that particular mesophil cells 2800 times, then their size comes out to be 5 micrometer in diameter. So option number B is correct here. The drawing has made from a section showing the part of the alveolus and red blood cells in the capillary. The magnification of the drawing is 5000 times. So here we have shown on particular alveoli and the red blood cells. So if the distance between them will be 1 micrometer, then they will be the best of the diffusion of the oxygen from the air into the red blood cells. A student calibrated a scale on an eyepiece reticule in the eyepiece lens of a light microscope. The student was given a stage micrometer scale to use. The deviance of the stage micrometer was 0.1 millimeter apart, which data must the student collect in order to calibrate the eyepiece reticule, right? Magnification of the eyepiece lens of the microscope, no, it is not required. Number of the deviance of the stage micrometer scale seen in the one field of the view of the microscope, no. The number of the deviance of the eyepiece reticule scale equivalent to the each deviant. We have to find out that particular deviant which is equivalent to the each deviant of the stage micrometer scale, right? So the option number D is correct here. The diameter of the living cells varies considerably. That of the eukaryotic cell is this. Prokaryotic cell is this one, right? So using these parameters, what is the maximum number of the each cell type which could fit along a line 1 centimeter long? So we have to find out the correct option here, 24. So number of the white blood cells, as we find out all these options, and we find out that the option number C is correct here, right? How many nanometers are there in the 1 millimeter? That comes out to be option number C, D is correct here, okay? The eyepiece lens of a microscope can be fitted with an eyepiece reticule. Which of these statements about the eyepiece reticules are correct? They measure the actual length of the cells at the micrometer? No. They help the biologist to draw the cells with the correct proportions? Yes. The change in size when the objective lens is changed from the 10 to 4? No. So the option number D is correct here because we find out that the second line is the correct one. Now we will move towards MCQ number 27. A student was asked to use the scale bar shown to calculate the magnification of a cell on a photomicrograph. It comes out to be 2 micrometer. Which method could the student use to calculate the magnification of the cell? Divide the diameter of the cell by the length of the scale bar, both measured in the same units of the length? No. Measure the diameter of the cell in the millimeters, multiply by 2000. No, it cannot be. The measure the length of the scale bars in millimeters, yes. Convert to micrometers and divide by 2, yes, it can be. So the option number 2 is C is correct here, right? So because we always measure the length in millimeters and convert it into the micrometer, so we have to convert it from the 2 micrometers, so we will divide it by the 2. Which eyepiece and objective lens combination of a light microscope allows the greatest number of the cells in the field of the view to be seen? So which combinations are the best one to view the greatest number of the cells in a particular slide? So we find out that the option number A is correct here, because if we use the 5 times eyepiece lens and the 10 times magnifying power objective lens, then we can view the greatest number of the cells in that particular sample. Which statements about the resolution and the magnification are correct? Resolution is the ability to distinguish between the two separate objects that are very close together. The number of the times larger an image is compared with the real. Yes. So I have read all the other options as well, and I find out that option number A is correct. An eyepiece reticule has a scale with 100 divisions. A stage micrometer has a scale with 50 divisions, each of which is 0.04 millimeter apart. Using a 40x objective lens, the whole length of the stage micrometer scale lines up with the 15 divisions of the eyepiece reticule. What is the actual length of the 100 division scale of the eyepiece reticule? So when we will calculate this, and then it comes out to be 13 millimeter. A prokaryotic cell, which is a 1 micrometer in diameter, is magnified 50,000 times in the electron micrograph. What is the diameter of the cell in the electron micrograph? So we will use the same formula here. Magnification is equals to image size by actual size. So magnification here is 50,000 by actual size is 1. So we have to find out the image size. Whenever we will calculate it, then it comes out to be 5 into 10 raised to the power 1 millimeter. Because we have the image size in micrometer, we will convert it into the millimeter, and it comes out to be 5 into 10 raised to the power 1 millimeter. So option number C is correct here. Which of these statements about the light microscopy are correct? The greater the resolution of a light microscope, the greater the detail that can be seen. Yes. The greater the magnification of a light microscope, the greater the detail can be seen. No. It is related to the resolution, not magnification, right? Increasing the magnification of a light microscope up to its limits of resolution allows the more detail to be seen. Yes. The shorter the wavelength of the light used in the light microscope, the greater the detail. Yes, because we have to view the shorter things. So if there will be the shorter wavelength, then we can see the more detail with it, right? So we find out that option number B is correct here, because it contains all the 3.1, 3 and 4, right, which we used to be the correct. Which steps are needed to find out the xylem vessel viewed in a transverse section using a 10x objective lens? Convert from the millimeter to micrometer by multiplying by 10 raised to the power minus 3. No. Calibrate the eyepiece reticule using a stage micrometer on a 4x objective lens. No, it cannot be. Measure the width of a xylem vessel using an eyepiece reticule. Yes. Multiply the number of the eyepiece reticule units by the calibration of the eyepiece reticule. Yes, it can be, right. So the 3 and 4 points are correct here. So we will find out that the option number D is correct. A student made notes describing the photomicrographs of the 4 cells. Gray cytoplasm at the edge of the cell contains many black lines and the spores. Large white area in the cell. Large white area in the cell may represent the plant cell, may represent that particular large vacuole present in the plant cell, right? So it can be the plant cell. Gray cytoplasm contains many black lines and spores which fill the entire cell. They can be the ribosomes. Pale blue cytoplasm surrounds a single dark blue spot. It can also be present. Dark blue spot can be the nucleus. So many green structures are enclosed. Green structures, so they can be in the plant zone. So we have to view all the options and we find out that option number C is correct here because it fulfills all the requirements. The diagram shows a slide of the transverse section of a stem. The diagram is the same size as the actual size. So a student observed this slide using a light microscope at a magnification of 40x. The student made a plan drawing of the stem which was 20 cm in the diameter. The student labeled the plane transverse section of our stem 40x. Why this statement is not correct? Why? We have to find out the reason why it is not correct. We have to read out all the options and we find out that the option number C is correct because the image size is the drawing was larger than the 40x, because 40x is not that much magnification power which can be viewed, which can view 20 centimeter stem, because we have to require a large amount of magnification, larger magnification to view the 20 centimeter diameter. A specimen of the plant tissue is first observed under a microscope using a red light with a wavelength of 650 nanometer. The same specimen is observed under the same conditions but using the green light, and its wavelength was 510 nanometer. What happens to the magnification and the resolution when using the green light compared to that of the red light? So in this case, I have read all the options and I find out that option number D is correct because the magnification remains same in both cases and the resolution increases. Which statement explains why it is necessary to use an electron microscope to see the chemistry of a mitochondrion? The magnification of the electron microscope is greater than that of the light microscope. The membrane of the crystal are separated by distance greater than the 200 micron meter. The resolution of a student microscope using a daylight is too low, and the wavelength. I have read all this, but I found out the C is the most accurate one because the daylight resolution is very much low, so we can't see the crystal in that particular daylight. A prokaryotic cell is about 0.25 micrometer in diameter and is magnified 50,000 times on an electron micrograph. How big will its diameter be in the electron micrograph? We have to find out with the same formula. Magnification is equals to image size by actual size. So magnification here is 50,000. Multiply it with 0.25 as we have to find out the final answer in millimeters, so we will divide it by 1000 and we find out that the actual image size comes out to be 1.25 into 10 raised to the power 1 millimeter. So option number C is correct here. When making the measurements in the experiments, which method could have the parallax errors? Parallax errors means which can occur side by side. Using a calibrated eyepiece reticule to measure the length? No. Using a measuring cylinder to measure the volume? Yes. Using a ruler to measure the length of the shoot? Yes. They can make the errors because we cannot use cylinders in that particular microscopic measurement. So the option number D is correct here because 2 and 3 lines are we can see correct in this particular case. A light microscope is used to observe the two structures that are 200 nanometer apart. When we will view that particular structures with the 40x or the 400x magnification power, then whether the distance remains same and when it will change, it will remain same. It will be about 200 nanometer, right? So option number C is correct here. So I will finish my lecture here and in the next lecture we will cover the remaining MCQs. Till then, thank you. Allah Hafiz.